Welcome friends. We will continue our discussion on reproduction in flowering plants. Now this is part 2. In the last episode, we have completed up to the production of pollen grains. And now from pollen grain onwards, we will continue. So, we have seen that in the microsporangium, some sporogenous cells are there. And these sporogenous cells, they are diploid. Now we took this a single one, and it underwent two successive divisions. The first one is meiosis and the second one is mitosis. So 4 microspore cells were produced, and these were initially they were like this. This is known as tetra, 1, 2, 3, 4, tetrahedral arrangement. So from this tetrahedral arrangement, we have come across in the last episode that 4 haploid nuclei are produced. These are microspores, and they have n number of, they are haploid, all are haploid nuclei. Okay. So these were, we told them that microspore. These microspores, up to this we have completed. And then we took one of these, not 4. We took one of these and say that this is pollen grains. This is pollen grains. Initially, it had a single nucleus with chromosomes and others. So this is pollen grains. Now we will go through the structure of the pollen grains, and we said it that there was 2 cells there, that will come in. We are coming. So this is a single nucleus with a cell. Pollen grains have 2 layers. The outer layer is like this, is a thick one. And this is inner one. The outer one we used to call it exine, and the inner one we call it intine. So the pollen grains have 2 layers. The outer layer is known as exine, and the inner one is known as intine. The exine is made up of, it is hard inside, it is thick and hard. It contains sporopollenin. Very important terminology usually comes in short question. This is actually the most resistant biological compound known, sporopollen. It is made up of sporopollen. It is thick, hard, and it is resistant to anything, because when these pollen grains will fall, it will be transported by wind, by flies, by kites, or whatever it is. So the pollen protects the inner microspore. Clear? So this is very precious, this microspore, that this precious, they are protecting it from two layers. That is, the outer layer is made up of sporopollen. It is the hardest and most resistant biological material known so far. And this intine is made up of cellulose. So these two we will write that the pollen grains are made covered by two layers. The outer layer is exine, inner layer is intine. Outer layer is made up of sporopollen and inner layer is made up of cellulose. This is the hardest biological material known so far. Now, in the outside, I have made so many pores. These pores are known as germ pores. Clear? So this is known as germ pores, or this can be called slits. S-L-I-T-S. These 2 points you should write. That in some places of the extine, although it is the hardest and although it is the most resistant, but it has some pores, minute pores. That minute pores are known as germ spore or slit, from where these will produce pollen tube. Pollen tube will come out and will fertilize the egg. Okay, this much we will write. Now we will go. This is not male gamete. Till now we are not saying that this is a gamete. We are saying that it is microspores. These microspores undergoes division. Now this microspore I will write. I will draw this. This microspore will be like this. This is intine. Exine is there. This is intine. And now this nucleus, 4 haploid nucleus, these nuclei will undergo 2 division. And it will produce 2 cells. And it will produce 2 unequal cells. The bigger one and the smaller one, the nucleus will divide. Here there will be one nucleus. Here there will be nucleus. Although this is a mitotic division. Now this will divide, this microspore nucleus will undergo mitotic division and will produce 2 cells. Clear? Here there is cytoplasms are there. A smaller cell is known as generative cell. Clear? And the outer, the bigger one is known as vegetative. V-E-G-E-T-A-T-I-V-E, vegetative cell. generative cell. Clear up to this? So the microspore nucleus will undergo mitotic division and will produce a bigger cell and a smaller cell. This smaller cell will remain in touch with the cell wall. The smaller cell will remain in touch with the cell wall. This is generative cell or generative nucleus, you can say. This is vegetative nucleus. Okay. Let it be nucleus. To be more precise, generative nucleus and vegetative nucleus or cells. Anything you can say. Now in the next phase, this remain attached with the cell wall. Initially, this is a smaller one. This nucleus and this cell will soon detach from the cell wall. Will soon detach from the cell wall, and it will be like a lens. Like a lens, and this portion will be there in time. This is in time, and X sign is there. So it will detach from the cell wall, and it will get free. And there the vegetative nucleus or vegetative cell, make it nucleus, vegetative nucleus will be there. And here there is cytoplasm. This portion is very important. Please pay your attention. So the generative cell will get detached from the cell wall, and it will float inside the cytoplasm of the vegetative cell. Clear? So this is the vegetative cell, the bigger one is entirely is vegetative cell, and this cell, which will look like a lens, will be floating in the cytoplasm of the vegetative cell. This point you should write. What I am saying, I repeat, that after the formation of two cells, vegetative nucleus or vegetative cell, generative cell, this generative cell immediately detach with the cell wall and will be a lens shaped cell having cell wall, and it will float within the cytoplasm of the vegetative cell. This much you will write. Now we have produced two things, that is here this one, and from this onwards, at this two cell stage, the pollen grains will go out. How? The anther is like this. Everything is happening in the anther or microsporangium. So this anther at this two cell stage of this microspore, the anther splits longitudinally. It splits like this. So not transversely, it will split longitudinally, exposing or dehiscing or ejecting the microspores. Ejecting this two cell stage will flow. Will flow in the air. If you had been to Himachal Pradesh and others, there you will see the pollen cloud used to come, and some have pollen allergy also. So these pollen grains will come like a cloud. So these pollen grains now go and fall into the stigma of female flower. If it is bisexual flower, it may flow in the, it may touch in the stigma of the same flower. If it is others, it will go there. So the fertilization will start. Now on longitudinal splitting, as the anther grow, as the microspore has generated two cells, and usually the anther passed longitudinally and ejecting the microspore outside. This much I will keep, and the growth of this will come whenever we go to female gametophyte, because until and unless it falls on the stigma of the female flower, it do not grows. Whenever it will grow, let me say a little bit, whenever it will grow, the intine will protrude. The intine will protrude outwards, and in that case the vegetative nucleus will be here, followed by male gamete, followed by male or with generative nucleus or generative cell. The generative cell will further undergo division, that I am coming. So usually whenever it falls on the stigma of the another flower, the intine protrudes out through the germ pores or slits and produces pollen tube on being put into the stigma of female flower. So this much is the male side. This microspores. Male are called microspore and females are spores are known as megaspores. Okay? This much is there in the male gametophyte. Now we have to go to female gametophyte. In female gametophyte, this is known as thalamus. Where ovary style stigma will be there. Okay. Ovary style and stigma. This is known as ovary style stigma. This is ovary style and stigma. This is the female flower. We will not go into the how it is produced, that we have already discussed. Now, in the reproduction, male part is over. Now, female part. The female starts. The female gametophyte starts as a protuberance, in the placenta or ovary. In the placenta or ovary, there remains a protuberance with numerous cells. With numerous cells, there is a protuberance. Clear? Then, sometimes in the examination it is asked structure of an ovule. Now, we are completing the same at a single shot. So, we will say that the female gametophyte is produced as a small protuberance on the ovary or placenta. This is known as, this is filled up with cells called nucellus. These cells are known as nucellus. It is a protuberance. Clear? This nucellus is growing. It grows. This will ultimately form embryo. That embryo will produce seed and it will be here. It will be here that as a seed. So this outgrowth is known as, is full of cell known as nucellus. This nucellus, now it will undergo so many divisions. There are numerous cells called nucellus. A hypodermal cell of the nucellus enlarges and becomes a bigger one. In the next phase, an hypodermal cell of the hypodermal, epidermal and hypodermal. Hypodermal cell of the nucellus get enlarged and bigger with prominent cytoplasm, distinct nucleus. This is known as archisporial cell, or this can be termed as microspore mother cell, which I am coming. This is megasporangium, and this is, we will call it, because in some books it is written, here also it is known as archisporial, archisporial cells. Cell. Or you may call primary archisporial cell. Clear? These primary archisporial cell will form an outer parietal layer. This is outer parietal layer, and inner sporogenous cell. So we can, the same thing as per this, I will call outer parietal layer and inner sporogenous cell. So the archisporial cell will undergo will enlarge and produce outer parietal layer and inner sporogenous cell or inner sporogenous cells. Here, this is sporogenous cell. This will be called as megaspore mother cell. I am coming. This primary sporogenous cell These primary porogenous cells or inner porogenous cell will act or functions as a MMC, megaspore mother cell. This inner sporogenous cell will function as a megaspore mother cell. That is megaspore mother cell, that is female gametophyte we are studying. This is written in one book. In another book, sometimes the primary archisporial cell, without getting divided into outer primary parietal layer or inner sporogenous layer, they directly act as a primary archisporial cell, can directly act as a functional megaspore mother cell, MMC, megaspore mother cell. These are the two thoughts. Somewhere it is written that this archisporial cell directly functions as megaspore mother cell, or in one book it is written that it may divide like the male gametophyte into outer parietal layer and inner sporogenous layer. The inner sporogenous layer acts as a megaspore mother cell, or sometimes it do not, this archisporial cell do not get divided into this outer and inner, they directly functions as megaspore mother cell. Clear? As this proceeds, as the enlargement, as the growth proceeds, then what happens? This nucellus, this is ovary, this is ovary, this nucellus raised at a stalk-like structure. And it forms an outer covering and an inner covering. This is outer covering, this is outer covering, this is inner covering. So, as the growth proceeds, as the female gametophyte proceeds in growth, the nucellus is raised on a stalk-like structure. This structure is known as funiculus. Clear? This place is known as raphe. And these are called the integuments. Integuments, that is covering. This is outer integuments, and this one is inner integuments. What are they? Integuments. Inner and outer integuments are produced, and as the growth continues, it raised in a stalk-like structures with numerous cells. Here, leaving a opening in one side. This portion is known as funiculus, this is known as raphe, and outer covering is known as integuments. There are two integuments, and this opening, it leaves an opening, it grows, and in some books it is written like this, and in some books it is drawn in this way, but this will be a better one, I think. So this is a opening this opening, you know, this opening is micropyle. Clear? Leaving an opening in the micropyle to allow the pollen tube to enter. This is acts as a passage for the pollen tube to enter. That we have given up to pollen tube, and the pollen tube will germinate, this will germinate, then fertilization will come. It is not that microspore mother cell and megaspore mother cell are produced. This megacell mother cell are 2n in number. This is 2n. And microspore, that is n, we have already produced this. We are coming that how it will produce a single. Now, this is micropylar end, and the opposite to this will be termed as chalazal end. The terminology. So this is known as chalazal end, this is known as micropylar end. And inside there are this functional MMC is there. I am coming. So in the next we will draw you can draw it in single A, but here there is no space, I am taking it here itself. So in the next page we will see that this is outer, this is inner micropyles. Here, what we see that here the megaspore mother cell, it may be divided into these, it may not get divided into outer and inner, but somehow or other they are functions as megaspore mother cell 2L. So you can write that come what may, but we are getting the megaspore mother cell. The megaspore mother cell here divides. It divides into two. The megaspore mother cell is here. This megaspore mother cell will divide into two by meiosis. Clear? So in the meiosis, first they will divide into two, that is N and N. This is N and N. Apply. Followed by, this is known as dyad. This is known as dyad. And followed by transverse, this is transverse division. So transverse division, this is there is two cells. Being meiosis, it will be haploid. Then again it will divide into two. One and two. This is N, this is N. So there are, this is known as dyad, and these four are known as tetrad. The megaspore mother cell, after you have written chalazal end, micropylon end, integuments, raphi, funiculars, then you will write that the functional megaspore mother cell 2n, these 2n will undergo meiosis, reduction, division and will form two cells which subsequently form another two cells and they stay in a linear manner. So this is known as linear tetrad. But I should say linear, Linear, linear tetrad. And what we have seen in microspore germination, it is tetrahedral. I have said you tetrahedral arrangement like this. 1, 2, 3, 4. This is tetrahedral. And a common one. Here there are 4. This is linear tetrad. This is the difference. You should write, pinpoint this information. There is tetrahedral arrangement. Here also it is linear tetrad. 1, 2, 3, 4. Clear? Now out of this 4, the magic is that in case of here, each and every nucleus undergo processing and becomes microspore or pollen grains. But in this case, the 3 disintegrates. 1, 2, 3. Here, these 3 disintegrates. So we should allow this in this way. This is outer, this is inner, and this cell will be a bold one, and these 3 will be disintegrated. So one haploid cell will remain. This is known as functional. You write this functional megaspore. This is functional megaspore. So out of these 4, 3 is not generating anything. So this method is known as monocarpic. Because only one grows, the 3 disintegrates. So this is called monocarpic method. Now in the next phase, these 3 disintegrates and 1 remains haploid, 1 haploid in remains here. Now, this will undergo divisions. This I am erasing, I am writing here. In the next phase, this is outer integuments and this is inner. Oh, one thing I have not mentioned. You should write the megaspore, the 4 megaspores produced following meiosis and mitosis. Only 1 remains active, that is active megaspore or functional megaspores, usually in the chalazal end, not in the micropylon end. Specific, you should be more specific in writing this, that in the functional megaspore, within bracket I am writing, you should write chalazal end. So at the chalazal end, this survives. This will undergo processing. Okay. Now here, in this area, these undergo again division. This undergo division, these end circles, it is not the bigger. Let me see. This is inner integuments, outer integuments, and only one functional megaspore is there. It remains encircled, and now the cell wall develops. I am coming. This megaspore now will increase in size. This will increase in size like this. With the cell nucleus, this will increase in size. So, increase in size. Megaspore functional. That is haploid. Still, it is not known as gamete. Now, what will happen? You see, these will undergo mitotic division. Clear? So, in mitosis, we know that two cells are produced. One cell will be at the chalazal end, and another nucleus will be at the micropylar end. So this will, after this, you should write mitosis. During mitosis, two cells, two nucleus or gametes are produced. So two nuclei has been produced, clear? And they goes to respective pole. This is north pole, this is south pole, or this you will say this is chalazal end, and another one goes to micropylar end. And this is known as embryo sac. Clear? Write it. Embryo sac. Space is restricted, otherwise I could have kept all the figures. This is known as embryo sac, and this is chalazal end, and this is known as micropyle, and this is stalk, and this is hypothalamus. You should draw this also, otherwise you may confuse. So this is stalk-like structure, funiculus. This embryo sac is produced. That is female gametophyte, not gamete. And its nucleus undergoes mitosis to produce two nuclei in the both pole. Again, there is very specific. You should draw another. I am doing it here itself. You should do another. It forms individually. It forms two cells here, two cells here. Again, it underwent or undergoes further mitotic division to produce two nuclei. So there are four nuclei from the single nuclei here itself. Clear? There are four nuclei. Again, it will give rise to two more nuclei. 1, 2, 3, 4. Here also it will be 1, 2, 3, 4. So on each side there are 4 and 4, 8 nucleated stage. This 8 nucleated stage embryo sac. Within the embryo sac it is from the single functional motherspore, megaspore, it is being produced. Now out of these four, they will sacrifice. They will sacrifice one. One will go move towards here. Another will move here. One will go here and another will come here. So these from the micropylar end, one nucleus will come to the middle of the embryo sac, another will go from the micropylar end, from the chalajar end, two nucleus will come here. These are called, actually these are called polar nuclei because it is coming from the pole. When they fuse, they come and they fuse. They fuse. Then this is called fusion nucleus. So this is called fusion nucleus or secondary nucleus. And here how much will remain? 3. One gun, one sacrificed, goes to the middle of the embryo sac, not the, total ovule. Only in the embryo sac, these 3 and these 3. These are called, it is known to you. These 3 are called antipodal. Antipodal cells. These are called antipodal cells. And here itself, here in the micropylar end, out of the 3, one will be bigger one, and it will be protected because this is the main female gamete. Only one female gamete. Here male gamete have been produced so many. There are only female gamete. It is very precious. They are protected by 2 security guard. These are called synergids. SYNERGIDS. And this is known as OVU. Clear? So this is the one protected by 2 SPG, special protection group guard. This is important in female gamete. Now this is gamete. This is known as gametophyte. Now this female gamete has produced, this fusion nucleus or secondary nucleus produced, antipodal cells are produced. In the next phase, we will go to fertilization. Because we have already prepared that one, that is very little, I will not take much more time, although it is more, or we can go to the next episode also. We will do the fertilization process. Till then, take rest and thank you very much. And please do subscribe